Okay, I'm starting to share my screen. Any questions?
No, I don't have any questions. I wasn't sure if we were doing a Q&A or going
What?
through
If we're doing what?
a Q&A session or going through the lab.
We're going through the lab today.
Okay. No questions here.
Okay. All right. Today we're doing lab 9. Today is Tuesday, November 9th. Please note that on November 11th, it's Veterans Day, or there's no class because of Veterans Day. So we'll not be meeting either in the lecture or the lab on November 11th. Any questions about that? All right, so today we're doing lab 9. We normally do the labs on Tuesday. Lab 9 is the identification. Are you seeing my screen? I should ask that.
Yes.
Okay. Lab 9 is the identification of gram-negative bacilli. You'll complete lab 9, work on some case studies, and there's a worksheet for lab 9. Please do the reading. It's not very much for lab 9, just a little bit of chapter 5 and chapter 10. Upon successful completion of this lab, you should be able to interpret the results of the following biochemical tests. The IMVIC test, which is actually 4 different tests, microbiological tests. IMVIC stands for I, indole, the M, methyl red, V for Voges Proskure, the I is just in there for a vowel, and then the C for citrate. So we'll talk about all those tests today. We're also going to talk about carbohydrate fermentation, which most of you know about because we've done that in the lab. We'll talk about the motility test and the hydrogen sulfide production test. Be able to explain how to interpret TSI test, that's triple sugar iron. We grow it on a slant. Be able to explain the SIM deep test. It's in a tube that's flat across, so we call it a deep. SIM, sulfur, indole, methyl, not methyl, motility test. And growth on McConkey's media and hectone enteric agar plates, or HE plates. You grow on McConkey's and HE plates to get gram negatives to grow, and it selects against gram positives, so only the gram negatives are supposed to grow. There are some exceptions though. And then you can use these 2 plates to look at lactose fermentation. And then lastly, be able to use the biochemical analysis to solve case studies involving gram-negative bacterial infections. So members that we're going to look at are 2 families, the Enterobacteriaceae family and lower down the Pseudomonaceae family. The members of the family Enterobacteriaceae are all small gram-negative non-spore forming bacilli. They are facultative anaerobes. There are 53 genera of Enterobacteriaceae. 24 of the genera are known to cause human infections. They are all glucose fermenting, oxidase negative organisms. And here are a table of some of the common members of the Enterobacteriaceae family that cause disease, like Salmonella and Shigella, you're probably familiar with. Escherichia for E. coli can cause disease. It is also part of our normal microbiota. Oops, wrong button. Most of these genera are normal microbiota of the intestinal tract, aka we call those coliforms, bacteria that grow in the intestinal tract. They are all opportunistic pathogens in that they can cause urinary tract infections, wound infections, pneumonia, and septicemia. Common genera responsible for causing opportunistic infections include Escherichia coli, more commonly called E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. You probably heard of Klebsiella pneumoniae. Several genera are associated with gastroenteritis. These include the genera Salmonella, Shigella, and then certain variants of E. coli, such as Serovar 0157H7. Pseudomonas aeruginosa is similar to the family Enterobacteriaceae in that they are gram-negative bacilli. However, the Pseudomonas family are different. They are in a different family, obviously. They're not in the family Enterobacteriaceae. They're in the family Pseudomonaceae. And they're all non-glucose fermenting bacteria, and they're oxidase positive. So that's two ways you can tell Pseudomonas from Enterobacteriaceae. Pseudomonas aeruginosa is an opportunistic pathogen causing infections, particularly in immunosuppressed individuals, burn patients, and trauma patients. Pseudomonas aeruginosa is a healthcare acquired infection, or another name is nosocomial infection. Pseudomonas aeruginosa can cause many infections in people, but it's lousy at getting started, and it is lousy at getting through the first line of defense, like your intact skin or your intact mucous membrane. You have to have that first line of defense broken or damaged before Pseudomonas aeruginosa can get established. It's important for microbiologists to identify and perform antibiotic and other biochemical testing to identify gram-negative bacilli that are cultivated from a clinical specimen. In this lab, we're going to talk about the common biochemical tests used to identify gram-negative bacilli. The first test we're going to talk about is one of the IMVIC members, the indole test. Some organisms produce the enzyme tryptophanase that converts the amino acid tryptophan into the products indole and pyruvic acid. After overnight incubation, Kovacs reagent is added, which reacts to indole to produce a red color at the surface. E. coli tests positive for indole production. And that doesn't look very red, but that's this photograph. That's positive, that's negative. Normally it's a red color. Tryptophan is an amino acid, and the conversion or the presence of tryptophanase is converted into indole, which causes the red color, and it's also converted into pyruvic acid. You only see the red color when you add the Kovacs reagent, and Kovacs reagent is a lipid-based, and that's why it's floating on the water. Two other tests in IMVIC are methyl red and Voges Prescure tests. They are two separate tests, but they use the same media, and usually we inoculate one tube and then divide that tube into two after they've incubated and run methyl red on half the tube and Voges Prescure or VP on the other half of the tube. Is that clear? Both MR, the methyl red test, and the VP, the Voges Prescure test, are looking at what products are made from glycolysis. If acid products are made, we use the MR test to look for the acid products, and we use the VP test to look for the neutral products. So glucose in glycolysis is converted into pyruvate, and the end products, other end products, can be acid. MR test will test for that, or they can be neutral products, and the VP test can test for that. So you inoculate an MRVP broth for 2 to 5 days. MR, you inoculate for 5 days. You can test it at 2 days, but if it is negative, you have to repeat that test and grow it for 5 days. And so we will just, in our lab, always use 5 days for methyl red. VP can be done at either 2 days or 5 days. So you add methyl red, a dye, to the product of the methyl red media that has a bacteria growing in it. And if the dye remains red, it then indicates a positive result for the methyl red test. If the dye switches from red to yellow, then it is negative. Let me show you the colors down here. So there's methyl red positive, and then VP negative. Voges per scure, you need to add the reagents, Barrett's reagent A and Barrett's reagent B to the tube, and then shake the tube to add oxygen, and then let it sit for 60 minutes. If acetone is present, it will turn red, at least in part of the tube. It may not be the entire tube. If it is negative, the tube will stay yellow, but you do need to let it go for up to 60 minutes. Usually the red color develops by 30 minutes, but occasionally I've seen it take as long as 60 minutes before it turned red. Any questions about the MR or the VP test? Like I said, you usually inoculate one tube and then divide it into two, running MR on half the tube and VP on the other half of the tube. The last test of the IMVIC test is the citrate utilization test. Here we're testing the bacteria to see if it can use the sugar citrate as its only carbon source. If it can use citrate, then it'll grow in the tube and turn the tube blue. If it can grow in the tube and use citrate, it'll also use ammonium, and it's probably the ammonia being produced that turns the tube blue. If it cannot use citrate, it will not grow in the tube and the color will stay green. E. coli and Shigella can metabolize citrate, so they will turn the tube blue. Staphylococcus epidermidis, if I'm remembering correctly, does not, don't hold me to that, I might not be remembering correctly, does not metabolize citrate, and so the tube will stay green. The tube is normally green, and if basic products are made, the tube will turn blue. So blue positive, green negative. Any questions about the citrate test? Oh, this is showing you the Krebs cycle, and citrate is a sugar in the Krebs cycle. If the bacteria can use citrate, it'll then run the Krebs cycle and grow. If it can't digest citrate, because citrate is the only carbon source, it will not grow. All right, carbohydrate metabolism tube is one sugar in a media that is fermented. And the tube is either orange or reddish or even a pink reddish. And then you put the bacteria in it. And if the bacteria can ferment that one sugar, it will become acid, and that will change the color to yellow. There are different sugars you can test for carbohydrate metabolism test. You can test glucose, lactose, sucrose, maltose, inulin, and I forget the fifth one, sorry. Or I guess the sixth one, because I said inulin, so that would be the fifth one. The tubes have an inverted Durham tube in it. That's that tube there that is filled with the media. And so not only are you looking at the production of acid from the fermentation of that sugar, you're also looking at the production of gas from the fermentation of that sugar. And if you have acid and gas, you'll see a tube like this, yellow with the bubble. If it's just acid, it'll only be yellow, no bubble. And if it's no fermentation, it'll stay pinkish or reddish. Any question about carbohydrate metabolism? All right. All of the Enterobacteriaceae family members can ferment the sugar glucose. Not all of them ferment producing gas. Some of them ferment and are acid only. Others are acid and gas. Pseudomonas aeruginosa, it will be negative for carbohydrate fermentation test. I should say glucose fermentation test. All right, the next the next test we want to look at is the motility media. This is a semi-fluid media. It does have agar in the tube, but the agar is very low in percentage. Normally the agar is at like 1%, and in the motility media, the agar is something like 0.4%. I'd have to look that up to be certain, but it's very low, and that makes it a semi-fluid media, and bacteria can swim through the motility media. And what you do is you inoculate with a needle the bacteria in the tube. And if the bacteria are non-motile, they'll only grow in the stab, which is negative for the motility test, shown here. If they are motile, they'll flare away from the tube. And let me draw you a picture. If I can open that. No. So here is the tube. Make it nice and wide. And there's the stab. If the bacteria grow out from the tube, and they usually are, I don't know, buffered, mountainous, usually rounded. And they'll be on this side too. If they flare out from the stab, then they are motile, slightly motile. If they are very motile, where's my picture? Then you'll see them all over the tube. And you can see that the tube is hazy. That's from bacteria growing in the media all over the tube. So this area you can see through the media. You can't see through the media here because it's hazy. And that's because this bacteria is very motile. If it's only slightly motile, it'll be flaring like this. Any questions about that? Anyways, that's the motility test. If the bacteria is motile, it has flagella, at least one flagellum. All right. We can also use the hydrogen sulfide test. This looks for the production of hydrogen sulfide gas. And you can use more than one tube to test for the production of hydrogen sulfide. You can use TSI or triple sugar iron, sulfide indole motility or the SEMS test, and you can use the hectone enteric auger plate to look for the production of whole hydrogen sulfide. In all of these 3 tubes testing for hydrogen sulfide production, the tube will get a black precipitate, or the plate will get a black precipitate if a hydrogen sulfide gas is produced. Some hydrogen sulfide producers include Citrobacter, Salmonella, and Proteus species. So this looks like SEMS, and the tube was stab inoculated. And this is hydrogen sulfide negative, hydrogen sulfide positive. All right, any questions about hydrogen sulfide test? For triple sugar iron, this test can look at the production of hydrogen sulfide, in which case you stab inoculate the tube, and it should go down a little further than here, because hydrogen sulfide is usually only produced in anaerobic conditions. Anyways, they'll turn the tube black down here. But on triple sugar iron, we're looking at the production of hydrogen sulfide, the metabolism of the sugar, both anaerobically, and then we inoculate the slant of the tube, the triple sugar iron tube, and you put the bacteria on the needle and you stab inoculate, and then you get more bacteria on the needle and you put it on the slant. And then you look at the color of the tube aerobically and anaerobically, as well as the hydrogen sulfide production. These tubes have only 1% glucose and 1% lactose and 1% sucrose. They will turn yellow if they ferment a sugar. Obviously, the bottom of the tube will turn black if they produce hydrogen sulfide. And then you look at the fermentation of the tube anaerobically and aerobically. And the understanding of these tubes is complex. This one is negative for hydrogen sulfide production. It's not black. It's negative for the metabolism of sugar. It's why it's not colored yellow on the slant and not colored yellow in the butt of the tube. This one is colored black in the lower portion of the tube and red on the slant. This one's a little tricky. The black is from hydrogen sulfide production, so that's produced here. Whenever hydrogen sulfide is produced, it does ferment the sugar. And you can't see the yellow here, but because it's black, we know it was yellow, and then the yellow gets hidden by the black. And so this is an acid butt, meaning this is yellow here. So it's fermenting at least one of the sugars, and actually it's fermenting, well, I guess I can still say it's at least fermenting one of the sugars, and that sugar is either lactose or sucrose because it's black. Can it be glucose? I suppose it could be glucose. Sorry, it could only be glucose. And then it's red or uncolored here and yet yellow there. And that means that only glucose is fermented. Coming back to here, glucose is present in only 0.1%, but the other sugars are present in 1%. And what happened in this case is the glucose was fermented here. And initially, if we would have looked at it, like a few hours after we inoculated it, it would have been yellow. But the glucose got used up because it's at low concentration, and it did not ferment sucrose and lactose. And when the glucose gets used up, there's no more sugar to produce acid, and then the bacteria start digesting the protein in the media. The protein, when it's digested, releases ammonia, which is basic, and that'll change the color from yellow to red, or then eventually pink. And that's why tube 2, only glucose is fermented, because it is red up here and then yellow or black down here. And in tube 3, we're actually seeing red on the slant and yellow here. And only glucose is fermented. That's why the slant is red. And it is yellow in the blood, but because when the glucose is fermented, it produces acid. And anaerobically, the growth is slower than aerobically because of aerobic respiration. And so that's why it is yellow in the blood, but all of the glucose gets used in the slant, and so it turns red. Is that clear? I know that's confusing. And then if the sugar is used and more than one sugar is used, it'll be yellow in the butt and yellow on the slant. And it's yellow on the slant because it's 1% glucose, no, 1% sucrose or lactose. And so the sugar does not get used up by a day or two. And no hydrogen sulfide is produced because it's not black. We can say gas is produced because we have a bubble here and cracking of the media. All right, any questions about the triple sugar iron test? I know it is a complex test. If I ask you questions about this test, it'll be simple. I won't ask you about this condition, and I won't ask you about this condition. I may ask you what black means, but I will simply ask what yellow means and what red means. Okay? And if I ask you a more complicated question, I'll give you this table here, not the picture, but the table. Which will be a little bit more clear than this because the table will tell you. I guess I have to give you the tube, don't I? Yeah, I have to give you this, the picture and the tube table. All right. The sulfur indole motility test, that's more likely called the SIMS test, is looking at 3 different tests you're running. You're looking for indole production, and if it's red in the Covex reagent, which is added after you incubate it, it will be red on the top. So this is negative, that's negative, and these 4 are positive for indole production. You look at hydrogen sulfide production, and if the tube is black, that means hydrogen sulfide is produced. And then you stab inoculate this media, which is very liquid, meaning it's not, it's a, what do they call that now? Lost the name of it. A semi-fluid media. You stab inoculate, and if the bacteria grow everywhere, like here, or almost everywhere like here, then it is motile. If the bacteria only grow in the stab, then it is non-motile. Any question about that? All right, so let me see. This is negative for all of them. No motility, no indole, no sulfur production. This is positive for sulfur production, positive for indole production. And you can't exactly see it, but the black is everywhere and it's cloudy everywhere. So we know the bacteria is everywhere, so it's motile. This is positive for indole. It's not motile and is not hydrogen sulfide production. All right, any other questions? Okay, I mentioned that you can, on McConkey's and hectone enteric agar, use these tests to look for the fermentation of the sugar lactose. If the sugar is fermented, lactose is fermented on McConkey's media, you'll get pink colonies, and the growth around the tube, the colonies may be pink. That's from the fermentation of the sugar lactose, and it makes it acidic, which changes the media to pink. If the sugar does not ferment the sugar lactose, it will stay its normal color, and we're just going to say it's not pink. For the HE media, if the sugar lactose is fermented, it'll be salmon colored, an orangish, reddish color. If it does not ferment the sugar lactose, the bacteria will be their normal color, which will say non-pigmented or white in this case. And if the bacteria can produce hydrogen sulfide production, the colonies will turn black on HE plates. All right, any question about any of that? So you do lab 9, and you got some video clips to watch on carbohydrate fermentation, the SIMS test, the MR and VP tests. Remember, that's 2 tests, triple sugar iron test, and the McConkey's agar. You'll also have to view some of the website microbuzz. Click on this link here, and then click on the differential test to the left to expand the menu. You only need to click on the test. You need to complete step 1 of the worksheet for lab 9. Let me show you that. Oh, I'm no longer there. Let me get to it. So you need to fill in this table. Some of these you're going to study this week, like the carbohydrate fermentation, TSI. Oxidase, I think we talked about last week, but we talked about it earlier. Anyway, so you can look on microbuzz for getting the information for filling in the test. I got the positive result for oxidase on here. The cells, or the spot where the cells are, turns blue. The negative result, you got to put it in here. Okay, so fill in this table and look at microbuzz for getting the answers you need. Where are we? And then complete the case studies. I've got the case studies all here. Case study 1, read about the case where the patient comes in with an infection, and then the different tests they did. This one was grown on McConkey's. They did the gram stain. Get your results for those. And then the day 2 test. Oops, the table is broken. This is the SIMS test. That's the MR test, the VP test, the citrate test, and the triple sugar iron test. Get your results from those tests, and then tell me the species of bacteria that is causing the infection in case 1, and you write that in the worksheet. Right here. For case 1, these are the results you need to fill in, and then say what species is causing the infection in case study unknown number 1. So do that for the case studies. There's 5 of them, and those are shown in the lab module. Where are we? Here we go. Case study 2, a boy had a turtle. Case study 3, an accident victim. Case study 4, a burn patient. Case study 5, everyone got sick from eating at a local restaurant, and there was a party at a restaurant. All right. Any questions about the lab module? In the worksheet, there's also some questions to answer in part 2. Complete the following. What reagent must be added to detect the presence of indole? Fill that in, and then answer the questions. And when you have worksheet 9 done, submit it in for lab 9. All right, I'll be here at 7:09. I'll be here at least until 8 o'clock to answer any questions. But once the last student logs off, I'm going to log off. So get started on the lab. See if anyone's here. Yeah, Malia, you're the only one here. Let me stop recording.